since today is actually the last lecture of the series, unfortunately, but we'll be doing something else. We're going to be taking a quick break for a couple weeks, and then we'll be moving on to something else, but you'll hear more announcements about that coming up. Right. So first, jumping right into it, we're going to do biology practice. Last time I used Jack Weston. This time I wanted to use this resource. I'm doing this just because I wanted to introduce some free resources that you guys may want to use as well when you're doing your practice, just so you get an idea of what's out there, what each program offers. Out of the free resources, there's also a lot of paid ones that have practice pastors as well, obviously, right? So the link to this passage is also in the Google Drive that's linked in the chat, or you can follow along here. Graphs may be a little bit difficult to see, but I tried my best to make them visible and fit everything on here. All right, take a couple minutes. Okay. So moving on to the question, and I always encourage students to go back to the passage after they see the questions, just because you don't really know exactly what you're looking for. Some questions may not need it, like for this one, you probably won't need to go back to the passage that much, but for some, it may be very helpful. So the correct answer is actually a gap junction. Oh, sorry. So yeah, that's just a simple knowledge question, right? Gap junctions, because it mentioned CX43, kind of junction to CX43 form, mentioned its connexin, mediates direct transmission between neighboring cells. So in this case, it would be gap junction. Desmosome, hemidesmosome, I don't think I've ever heard these words ever like shown on MCAT before, so I highly doubt that you'll see them in a real MCAT. Tight junctions are a little bit different than gap junctions. Won't go too deep into the explanation because they're not really also that common, but just know that when you use gap junctions, which type of cells use gap junctions, which type of cells use tight junctions, and the difference between them, okay? But like I said, it's not really too common. Next question. Okay, so the correct answer is actually C. The inhibitor diffuses through the cell membrane. What do you know about this inhibitor? Well, it's made of a glycerol backbone, 3 saturated fatty acid tails. What does that sound like? That sounds a lot like a triglyceride. Well, because glycerol and 3 fatty acids sounds a lot like triglyceride. What is a triglyceride? That's a lipid. How do lipids usually get in cells? Well, what are some common lipids that are found in our bodies that we usually use? Well, one of them could be hormones like testosterone, cortisol, things like that. Those are common lipids or similar molecules. And how do they get into the cell? They just go right through the membrane because, again, the membrane inside the membrane is very hydrophobic. So something hydrophobic like a fat triglyceride or hormone molecule can just go right through, right? So that's why it's C. The inhibitor is recognized A and B. The inhibitor is taken up by a non-specific receptor or by a specific receptor. That would be more like a peptide molecule or something that's not some kind of signaling molecule, or something that's not necessarily like similar to a hormone, or something that is like a signaling molecule more than like a lipid or fatty acid. And D, the inhibitor passes through a cell membrane via channel. Mainly, the only things that pass through channels are ions. So this is not an ion, so it wouldn't be D. Okay. And this is the next question. Okay. So this one's a little bit tricky, so I'm going to spend some time going over it. The correct answer is A. Right. And let me explain why. I think for me, when I was reading through this question earlier today, I thought of it during using process of elimination. So we're deciding to interrupt this interaction, right, without using a small molecule inhibitor, which would most likely disrupt this interaction. Well, plasmid producing this would obviously not work because it would make just more of the CX43, so that wouldn't really make sense. Acetyltransferase specific for B-actin. Well, B-actin is mentioned, and I'll go back to it in a second. The passage B-actin is not really mentioned at all in the passage. B-actin was used as kind of a housekeeping molecule for one of the tests. I think that was the only time it was mentioned, but otherwise it's not really relevant at all. And radiation against heptanol, that's not exactly relevant either, so wouldn't be this. And so the correct answer, just by passive elimination, could be A, but the reason is because siRNAs are specifically can be used for doing this. And here, I'll go back to the passage. Right. So like I mentioned, I think this is the only time they mentioned B-actin, and if you notice here, it's just really just like a housekeeping gene, or it's just something that you know that the proteins that the cell is going to be expressing. So it's kind of like a control group, should be expressed in all three. And it's just kind of like to show the difference between sex 43 between the three groups. And then I believe they mentioned surgeries. Right, they used heptanol, a well-known inhibitor, to alter the function of CX43. So that's why radiation against heptanol wouldn't really work, because it's the inhibitor, so it doesn't really make sense. Why B would be the right answer, right? And they confirm the interaction through and really it was just by using process elimination. That's the main way, unless you know what siRNA is, right? Which is very rare, so I would expect most people wouldn't. In case you do, it was even simpler, but really you could do process elimination. Just to, this one doesn't make sense, this one doesn't make sense, this one doesn't make sense, so must be A. Okay. Okay, the correct answer is, for this one, is actually C. And let's go over why. So A and B, all expression control occurs at the transcriptional or translational level. First thing you have to recognize, transcriptional means the amount of mRNA will be different. So the actual transcription of DNA to RNA. And translational means RNA to proteins. That at that stage, the way we can actually look at this from this graph is we can see that this is actually measuring the mRNA levels, and this is measuring, this is measuring the mRNA levels, the graph's measuring the mRNA levels, and this is measuring the protein levels, these black bars, okay? So we noticed that if it was just transcriptional control, then we would see it as we do here. In here to here would be, there'd be a big difference, right? Because if it's transcriptional control, it gets changed at the mRNA level. If it was translational control, it gets changed at the protein level. So the amount of RNA is actually should be the same. So if it was at translational control, it would be, this would be exactly the way it is, except these three bar graphs would be about at the same level, right? And only this would be different, really, okay? If it was all transcriptional control, then this bar graph would be the way it is, but this would actually not be this way. It would actually all be, you wouldn't see that this effect, right? Because if it was transcriptionally controlled, you would have RNA, but then you wouldn't have the RNA to make the protein, right? Okay, that makes sense. So because you have both the bar graph different and this looking like this, between these two especially, right? They're the same. So it means that there's both transcriptional and translational control. So A and B are both wrong because it's a little bit of both, okay? Now I'll maybe go over that again afterwards, okay? But hopefully that makes sense. A and B can't be right because it's not all expression occurs at this level. The expression occurs at both levels. D, normal livers treated with heptanol have lower levels of CX43 compared to donor treated with hepanol. Well, where do you, it just says based on figure 1. So we're really just looking at figure 1. So normals compared to donors treated with hepanol, we're just looking at this graph. We don't really kind of see that at all. So it wouldn't really make sense for D, right? C is much easier to see. Donor livers have higher levels after reperfusion compared to normal liver tissue. This is normal liver tissue, right? This whole column. Actually, I'll do this. This is normal liver tissue, and treated with have higher levels of CX after reperfusion compared to normal liver tissue. This is after reperfusion, right? You can see the amount of mRNA is higher, amount of protein because of this band is higher. It's very simple, very easy just to see that. So see the correct answer, B, A, or not. Hepanol we're not really talking about right now. Not really. We're talking about reperfusion, and we're just basing it off figure 1 because that's what they told us to do. Okay. And again, expression occurs at the translation versus transcriptional level. It can be at the translational level only because you can see there's difference in mRNA. It can be at the transcriptional level only because even though you have differences here between this and this, these are equally as, because there's a difference between the mRNA levels, this and this has no difference, right? So because of that, there must also be some transcriptional control as well. Otherwise, these would be slightly different. These two graphs here, these two lines, how what they're called, sorry. Bands, these two bands. Okay, hopefully that makes sense.